everybody. Reporting to you again from the Glamour City, Hollywood.
Every patient who receives chemotherapy is receiving damage to their bone marrow because those chemo drugs, they kill rapidly dividing cells, and the bone marrow is the most rapidly dividing cell type. So their immune system is severely damaged. And a cancer patient needs their immune system because their immune system, as we know with the immune oncology revolution recently, is what's keeping their tumor in check. So the reason you and I don't have cancer right now is because our immune system is surveilling and eliminating these nascent tumors. This is primarily why cancer is an age-related disease, because the immune system fails to surveil it. And so one of the indications we're pursuing is patients going in for chemo. We isolate some of their bone marrow and we expand it, and then they receive the chemo. It eliminates some of their existing bone marrow. And then between chemo cycles or post-chemo, we reinfuse and reboot and restore their immune function. It's an enormous market. Every patient getting chemo.
Huge, massive.
Absolutely huge. And the holy grail is not only would it prevent the acute problems like infection while you're in the hospital getting chemo and, like bleeding and a lot of other complications acutely, but the holy grail and what I'm really hoping for is that it will prevent tumor relapse. So on a multi-year basis, if you've had cancer once, you're quite likely to have it again. Because if you've had cancer, it means that you're aging at a rapid pace and your immune system is failing to detect cancer. So by rebooting the immune system, hopefully it will prevent that risk that all cancer patients face, which is tumor relapse, either the same tumor or a different type of cancer entirely.
If you love listening to this show, please consider giving a rating and a review on Amazon Alexa or wherever you listen. We want to continue bringing you this amazing content, and part of our ability to do that means that we need a big audience. Now, it may not seem like much, but rating and reviewing the show will help more people find us, just like how you found this show. Simply on any podcast platform, search for a show, scroll down to the bottom, and push 5 stars. It's that easy. Thanks for supporting the show. Today, I'm joined by Sebastian Brunemeier, General Partner at Healthspan Capital and CEO of ImmuneAGE Pharma. Welcome to the show today.
Thanks for having me.
So, Sebastian, you've co-founded a number of longevity biotech startups, including Cambrian Biopharma, Samsara Therapeutics, and Cyclone Therapeutics. I wonder if you could just briefly tell us about each of those and where they are today.
Sure. So, my background is as a biomedical scientist by training. I spent 10 years in the longevity biotech field, first as in the lab as a scientist, and then as an operator and VC investor in the space. So some of the companies that I help launch, Cambrian is one of them. This is there in New York, your neighbor. It's one of the larger aging-focused multi-asset companies. Another, you mentioned ImmuneAGE Pharma, which is working on immune system rejuvenation. I can get into the details. Samsara Therapeutics, which is based in Oxford, UK, which is working on this process of autophagy. So when you do fasting and exercise, part of the reason it's good for you is it turns on this cellular process called autophagy, from the Greek self-eating. It breaks down the internal components of the cell and then rebuilds them. And that company's putting its first drug in the clinic in about a year. I also help launch Apollo Health Ventures, which is today the largest aging-focused venture fund. And today I run a smaller venture fund, Healthspan Capital, with some partners. And we are, as of last year, the most active investor in the longevity biotech or LongBio field. We did 15 deals. So yeah, that's my background in a nutshell.
So just to get a better understanding of some of the startups that you help launch, are you still involved with them? And what was the general role that you played in some of these entities?
Sure. So, I've involved in most of them. And they, to my great pleasure, are continuing to grow and be successful. The unifying theme of all of them is targeting the fundamental hallmarks of aging. So, there's been a revolution in the biomedical field over the last few decades where we can describe on a molecular level the damage that causes aging. And we have been able to extend healthy lifespan across species, like in lab mice, by up to 50% in mice through different genetic and pharmacological interventions that could be combined for synergy. And so, it's no longer a question whether we can extend healthy lifespan. It's now a scientific fact that the aging process is malleable. And it's only just started to become a big booming field with some billions of dollars coming into the space from fairly sophisticated investors, but we're still quite early. But, I was lucky to be there at the earliest stages.
So we've been referring to longevity biotech, but can you describe that a little bit more for the audience who are not familiar with the space?
Sure. So longevity biotech, or LongBio, is the world of interventions that slow the aging process and address the root cause of age-related diseases rather than the typical pharma approach, which is palliating the symptoms. So pharma tends to develop drugs that work very late in the process of disease. For example, if you have cancer, they will give you a chemotherapy that tries to kill rapidly dividing cells like cancer cells, but that's long after the process of cancer has been allowed to emerge. And so, what if we could understand the root molecular causes that cause cancer in the first place? Things like DNA repair, or the ability of cells to commit suicide when they realize they're becoming cancer, or autophagy, like I mentioned, or what I'm working on now with ImmuneAGE Pharma is rejuvenating the immune system. So the reason why hopefully we don't all have cancer at this very moment is because, it's always arising. Cancer cells are always arising every day, but they're being eliminated by these cellular and physiological quality control mechanisms like the immune system. The immune system is always patrolling for pathogens and for cancer cells. And so the LongBio field is asking the question, what are the root causes on a molecular level of aging? And aging itself is the primary driver of the most common killers today: cardiovascular disease, cancer, dementia, et cetera. So instead of intervening very late in the process and palliating the symptoms as the traditional pharma model is, what if we could intervene closer to the root cause? So the LongBio field has been growing rapidly in the last just several years. Some billions of dollars have come into the space, but it's still quite small compared to the incredible potential of the field.
Well, I think even just this week, there's been a lot of interesting news that's come up. I think there's a Harvard-affiliated entity that's come up with a new cell therapy approach to eliminate established tumors and induce long-term immunity, for example, to prevent cancer from recurring, to new forms of going beyond Cas9 that can start to attack DNA and RNA and rejigger things. So a lot of interesting things. Specific to ImmuneAGE Pharma, how are you guys developing therapeutics to rejuvenate the immune system with focus on aging of the bone marrow and the hematopoietic stem cells, or HSC, as a primary upstream driver for immune senescence and give rise to other blood cells?
Sure. So, this is the coolest time to be in biotech. The tools that we have are pretty mind-blowing and only increasing in their power. And a lot of capital is flooding into the field. So you mentioned that PACE technology out of MIT that could replace CRISPR, many groups working on alternatives to CRISPR, and then the gene and cell therapy field, which is another revolution. So to get into ImmuneAGE specifically, so we have a platform to discover drugs that rejuvenate the immune system at the source, the hematopoietic stem cell, or HSC. So HSCs are in your bone marrow. They give rise to the entire immune system. All of the white blood cells, T cells, B cells, even the red blood cells derive originally from these HSCs. They're sort of these superpowered stem cells. And when you do a bone marrow transplant, this is a reboot of the HSCs in the immune system. So, I've been a scientist, biotech VC, and operator in the LongBio field for 10 years now. And to my surprise, there has not yet been an effort to systematically identify molecules that rejuvenate the immune system. But we know that they exist. It can be done because of some serendipitously discovered molecules in academia that are known. Rapamycin is one example. And so we're bringing industrial strength muscle to immune rejuvenation drug discovery. And you kind of have to ask the question, why hasn't this been done previously? I mean, it's so important. The immune system is unique in that it's an organ system that travels all over the body and is responsible for policing it, pathogens like viruses, as well as cancer cells. And so, it seems like a really obvious area to really focus on. But part of the reason I think why this hasn't been done is because there was a missing piece of the puzzle, missing technology that we now have, which is it was not possible to systematically discover HSC rejuvenating compounds because the supply of aged human HSCs is relatively limited. They're very rare and precious cells. But one of our collaborators published in Nature a couple of years ago, his work at Stanford, now he's at Oxford, developed a method of expanding the numbers of HSCs by a factor of 1,000-fold. Prior efforts were about 10x. And so that allows us for the first time to do large-scale systematic screens for molecules that rejuvenate HSCs ex vivo, as we're doing in our labs in Switzerland. And so, we can also combine these reagents. So say we don't need to just stick to one molecule that rejuvenates them. We can have a cocktail approach. So I kind of joke we're sort of like bartenders or mixologists for these HSC rejuvenating cocktails. But that's what we're doing in a nutshell.
So I'm going to come back to the specific things that you guys are doing in terms of new method for expanding HSC. And I think there's a novel non-toxic small molecule that you guys have discovered. But I want to go back a little bit more. Again, this is just to help the audience kind of baseline and understand and get a good grasp of what they're listening to. When it comes to HSC transplants, that is really powerful because it treats, like I mentioned before, cancers as well as other immune system disorders. You can derive some of this from the bone marrow, but it also can come from peripheral blood as well as umbilical cord blood. So my first question is, why the bone marrow, and why don't we look at some of the other options?
Yeah, so good question. Umbilical cord blood, most people don't have their umbilical cord blood stored, but that's a great idea. People should do that for their children. Expansion of umbilical cord cells is limited, but our technology applies there too. You can derive HSC's CD34 positive cells from circulation, but they're relatively scarce, but we're working on that. That could be another advantage. Instead of having to aspirate from the bone, like the hip bone, a small amount of bone marrow, which is an outpatient procedure, can be done in 15 minutes, as compared to the full-on bone marrow transplant, which is under general anesthesia. You can get HSCs relatively easily. And the long-term vision of this is what we call the immune system reboot. So, don't tell my investors, but I'm not primarily doing this for the money. I'm in it for the therapies that will rejuvenate myself and my loved ones in the world. And doing an immune system reboot in the animals has shown to dramatically increase their lifespan over 15% when you transplant the bone marrow of young mice into old mice, and they're healthier in every way that you look. So how do we do that in humans? Well, the vision is we mobilize some of the HSCs in the peripheral blood, or we aspirate a small amount from the bone, put them in a dish, expand them, treat them with our proprietary cocktail of rejuvenating reagents, and then reinfuse them into the bloodstream, as is done typically in a bone marrow transplant. And the amazing thing about HSCs is they know where to go. They home into the bone marrow and repopulate it. So that is the vision, which is every some years, 5 or 10 years, everybody over age, say 35, will come in and just gently get a replacement or a tune-up of their existing clones, the existing HSCs in their bone marrow.
So I think you're referencing some of the journal articles that talked about transplanting young to old bone marrow that helped increase lifespan in mice by, I think, 12 to 28%. But again, there has been quite a bit of, I think, towards mid-end of last year, where even within the research community, they recognize that trying to extrapolate these clinical studies from small animals to larger animals and then eventually humans, it's not exactly the same thing. And then things that are promising with mice in the lab doesn't necessarily prove to be what it should be in humans. So given the fact that we're still very early in the process and we're not quite there in terms of later stage clinical studies, how do we know this is going to really pay off?
Yeah, I mean, this is the perennial question for all biotech and pharma, that translation from early results in the lab into the clinic. There are a lot of lines of evidence that support this approach. So there's a lot of human genetic data showing that HSC malfunction, it accelerates aging in other diseases. And non-obvious diseases too, like cardiovascular disease. One of the genes that most accurately predicts cardiovascular disease risk governs hematopoietic stem cell function. Furthermore, the pharmaceutical industry is not very efficient. It wastes a lot of money, in part because it's bureaucratic and it's too big and it's cartelized and not very innovative, but also because there's a fundamental philosophical problem with the way they do drug discovery. This is described at length by my friend Jack Scannell, who coined the term E-Rooms law. He's an ex-biopharma operator and equity research analyst, and he basically pointed out that it's getting exponentially more expensive for big pharma, not necessarily biotech, but big pharma, to develop new drugs with their internal R&D pipelines. And part of the reason he speculates, E-Rooms law being, of course, the inverse of Moore's law, part of the reason he speculates is because the animal models of disease that they use are not very accurate to the human case, right? So like if you have an animal model of cancer or, God forbid, Alzheimer's, what they call Alzheimer's with the amyloid beta mice, which is not very reliable, it ends up not working well in the clinic. And so it's the degree of contrivance of these animal models. The beauty of working with just normally aging animals as we do is that we haven't genetically engineered the animals to show some model of disease or administered some toxin or something contrived and unrealistic. This is normal wild type aging, similar to how it occurs in humans. And the aging process on a biochemical level is highly conserved and similar across many different species. So that's part of the reason we think longevity drugs that extend healthy lifespan across species are more likely to work in humans. And that's the empirical fact, which is drugs like rapamycin or metformin that extend lifespan in animals work for many different diseases in humans. So that's part of the reasoning. But again, we still have to do those clinical trials, but we're pretty optimistic.
Absolutely. Now, speaking of rapamycin, I think there was a recent article, I can't remember if it was Nature or not, that kind of debunked it. I think there was a couple areas that it debunked, rapamycin, or it was maybe one of the others, and then intermittent fasting, and that it didn't really have a statistically significant impact in anti-aging as we thought that it did. How do we reconcile some of these kind of mixed, results or studies?
Sure. So nobody's debunked rapamycin. Rapamycin extends lifespan across species very robustly. It's one of the best results we have. But people, I think metformin is one that is maybe a little overhyped. Yeah, you may have been referring to metformin, which is another popular drug. It's the most prescribed drug for diabetes. It lowers your blood sugar. And there's some evidence that it could extend human lifespan. I think that's primarily only in people who are metabolically unwell, and that's what the evidence seems to suggest today. So as far as intermittent fasting goes, I think the preponderance of evidence is strongly in favor of fasting. Intermittent fasting is a relatively newer area of study, so there may be some mixed results in the clinic, but I think the preponderance of evidence is in favor. I do intermittent fasting myself. And so, there may be some animals that don't do as well with intermittent fasting, but the mice do well with it too. So yeah, I mean, as with any area where you're having tons and tons of people doing research, you sometimes find conflicting results, but the preponderance of evidence is in favor of fasting for sure. In fact, for thousands of years, fasting has been recommended by most religions as a way to improve health and even cognitive well-being. Part of the reason is it induces ketosis, the formation of an alternative fuel source. So, if we had a drug that mimicked fasting or exercise, the benefits of fasting or exercise, it would be the best-selling drug of all time. And in fact, we've invested in drugs that mimic some of their benefits. So yeah, fasting and rapamycin are quite strong evidence, I would say. Metformin, in my personal opinion, is probably a little bit weaker, maybe a little bit overhyped. But there are also really, really interesting sort of new revolutionary interventions now, like transient reprogramming, for example. If you've heard of epigenetic reprogramming, this is relatively new. There was a Nobel Prize awarded for it in 2016 to Shinya Yamanaka. Basically, you can insert 4 genes into a cell, and it will reset the epigenetic age of that cell and reverse most elements of aging. And when you do that in the mice, it's a little more challenging, but we also do see restoration of function. The jury's still out on lifespan extension there, but in principle, if you could rejuvenate and reset the age of a cell, bringing it back to like an embryonic stem cell, day 0 age cell, as we can do in the lab routinely all over the world now, in principle, you could do that in the whole body because humans are just an agglomeration of cells. It's just an engineering challenge at this point of delivering those genes to enough cells and controlling them tightly to mitigate safety risks. But we have a bunch of groups working on small molecule mimetics of Yamanaka factors as well.
Well, I'm very excited, and you can't tell because I'm just not a very extroverted person, but sure. I'm very excited by where we are, and I think this is a very exciting tipping point. Before we get into the IA101 novel molecule that you guys have identified, let's talk a little bit briefly about the risk of HSC transplantation. Typically, while it's been very effective, it also has some potential complications. And I wonder if that's one of the other reasons why it hasn't really been actively pursued until recently.
Yeah. So the HSC, so bone marrow, there are tens of thousands of bone marrow transplants every year in the US alone. Each bone marrow transplant costs $500,000 to $1 million. And it's actually increasing in popularity because it's being used for more interventions. So for example, not a lot of people know this, but you can actually cure somebody of HIV completely with a bone marrow transplant if the cells that you're donating have a certain mutation in a gene called CCR5. So that's just one example of like the considerable potential of being able to reboot the immune system. Of course, bone marrow transplants are mostly still used primarily in leukemias. And the danger of the bone marrow transplant primarily does not come from the bone marrow transplant. It comes from preparing the patient by giving them high-dose chemo or radiation to eliminate their existing bone marrow, which is cancerous, to replace with alternative non-cancerous bone marrow. So that's where most of the damage is done. And we actually are trying to mitigate those risks to provide an alternative to that high-dose conditioning, it's called, of the existing bone marrow. There's another intrinsic to bone marrow transplant, or at least an allogeneic bone marrow transplant from another person, which is called graft versus host disease, GVHD. So in graft versus host disease, it's the grafted new bone marrow is rejected or attacks the rest of the patient's cells because it's recognizing these cells as foreign. We are focusing on autologous bone marrow transplant, which is your own bone marrow primarily. So there isn't that risk of GVHD. Although we think we can reduce the risk of GVHD with our methods in autologous or allogeneic bone marrow transplants. So, a big part of what we want to do is make bone marrow transplants, the safety per se isn't the problem. It's the availability of donor cells. And so expanding that supply opens new doors for many people to get bone marrow transplants or just even bone marrow supplementation that they wouldn't otherwise be eligible for. And so to give you one example, one of our investors, I was pleased to say, we're still early stage, but we have some pretty high profile investors, like, for example, Balaji Srinivasan from Andreessen Horowitz and Coinbase, as well as Thomas Ebeling, who is a former CEO of Novartis. And Novartis is one of the biggest oncology pharmas and one of the most sophisticated of all big pharmas based in Switzerland. And the area that he is most keen on, and I generally agree, is an indication, a use that is a very large market indeed, which is every patient who receives chemotherapy is receiving damage to their bone marrow because those chemo drugs, they kill rapidly dividing cells, and the bone marrow is the most rapidly dividing cell type. So their immune system is severely damaged. And a cancer patient needs their immune system because their immune system, as we know with the immune oncology revolution recently, is what's keeping their tumor in check. So the reason you and I don't have cancer right now is because our immune system is surveilling and eliminating these nascent tumors. This is primarily why cancer is an age-related disease, because the immune system fails to surveil it. And so one of the indications we're pursuing is patients going in for chemo, we isolate some of their bone marrow, and we expand it, and then they receive the chemo. It eliminates some of their existing bone marrow. And then between chemo cycles or post-chemo, we reinfuse and reboot and restore their immune function. It's an enormous market. Every patient getting chemo.
Huge, massive.
Absolutely huge. And the holy grail is not only would it prevent the acute problems like infection while you're in the hospital getting chemo and, like bleeding and a lot of other complications acutely, but the holy grail and what I'm really hoping for is that it will prevent tumor relapse. So on a multi-year basis, if you've had cancer once, you're quite likely to have it again. Because if you've had cancer, it means that you're aging at a rapid pace and your immune system is failing to detect cancer. So by rebooting the immune system, hopefully it will prevent that risk that all cancer patients face, which is tumor relapse, either the same tumor or a different type of cancer entirely.
So what you're saying makes perfect sense. And I think, for anybody that has had questions around the potential total addressable market, it is significant. And the reality is, when it comes to cancer, it's not a matter of whether you're going to get it. It just matters when, as long as you're living, right? The real issue underhand, and this is what you're trying to address, is the supply issue. So like you mentioned before, you yourself and other researchers that's affiliated with your entity were the first ones to create a new method for expanding HSC in ex vivo, achieving that 1000x expansion than any other prior known methods. And you mentioned before also the cocktail therapy, which is very critical for this as well. And it's this ability to be able to identify new molecules that can be applied in ex vivo to rejuvenate the cells in a dish, then transplant it back into a patient. And it's their own cells, they're not going to have immunity issues. That mitigates the risk of toxicity as well as the bad pharmacokinetics. Now, specific to the first molecule you guys have identified, which is the IA101, tell us about that. And then really what I'm interested in understanding is the really kind of the machine learning or the modeling you guys are doing to systematically identify other molecules that could also have incredible effects as well.
Yeah, absolutely. So that's right. So we have a two-part platform. So one is the HSC expansion. We call that stem supply. That could alone, be a major advance in the bone marrow transplant field. But in addition to that, we have a platform which is primarily based in Switzerland today for identifying new molecules that rejuvenate the HSCs. So we can screen at high throughput ex vivo outside of the body. And some of these molecules will even be suitable to take orally to rejuvenate immune function. And one of the first molecules we've identified with our academic collaborator is a molecule that basically is a metabolic reprogramming agent. So it turns over the mitochondria. So the mitochondria are the energy powerhouse of the cell. They do a lot of important things. They're critical for aging and also for intelligence and cognitive function. And they're very important for immune function because immunity is a very energy demanding process. And so we found a molecule that induces mitophagy, so the clearance of damaged non-functional mitochondria, while also inducing the creation of new mitochondrial biogenesis. And it works in a very interesting way that's different from most other molecules out there in the literature. And in addition to that one, we've found a couple more, including a bunch in the literature that work on all kinds of different pathways that can be combined for synergy. So we could really turbocharge and rejuvenate these HSCs. And we're seeing in the literature pretty dramatic results in the animals. So yeah, we're very bullish on creating this as like a multi-asset proper platform company that can take many shots on goal.
So just to be clear, on the previous identified molecules from prior studies, is that open source? Are there any patent issues, infringement issues?
Yeah, so for some of them, they're very well known in the literature, but they've never been taken into the clinic, or at least not for this indication. So there's marketing exclusivity there. There's intellectual property to be had there. We have a very strong medicinal chemistry group. And so we actually attempt whenever possible to improve upon the existing chemical matter in the literature. So for example, let's say we proceed with IA101, and then we throw in rapamycin into the mix, and then we throw in maybe an NAD precursor, and you name it. Some of those can be natural products existing as they do. Some of them will be proprietary new chemical entities created by our chemists. And the combination thereof is eminently patentable. It's non-obvious innovation. Perfect. So those cocktails themselves, even if we took completely natural products or generics off patent, just that combination alone is pretty strong IP. But we're also going a step further in making new chemical entities and identifying the targets that are not known for some of these molecules as well. So the IP strategy is, I'd say, pretty solid.
So some of the innovations that we're seeing, and certainly the startups as well, is trying to de-risk some of these different clinical stages. In other words, while it may look very promising preclinical, maybe even clinical 1, by the time it gets to level 3, maybe it just isn't there, and it becomes a financial disaster. So in that regard, again, this was kind of what I was referring to, is kind of the computational biology side. So while there's a lot of good assertions, good theories, but combined, you don't really know truly how it's going to really impact, right? So how are you modeling some of these? So talk about the computational aspects and the models.
Sure. So this kind of field lends itself to big data. So we're doing transcriptomics, single cell sequencing on HSCs and other immune cell types. By the way, I should mention that the molecules we find that rejuvenate the HSCs, they can also apply to other cell types, mesenchymal stem cells, which are used widely in clinical trials today, other immune cells like T cells or NK cells or macrophages, et cetera. So we have these large data sets, and we need to crunch the numbers, crunch the data. And so we're building out an AI ML capability there too. But I think the AI ML aspect, there's been a lot of hype in the drug discovery field for some years, and it hasn't panned out. Maybe now it will. I think there are very serious players in the field, like Google's new company, Isomorphic Labs, probably pretty talented. And one of my partners at Healthspan Capital is a Google AI guy too. So I think, we may be on the precipice of major breakthroughs in the AI drug discovery field. But the point you bring up is the key, which is the animal models of disease are not very reliable. So even if you have really good AI, pharma still relies pretty much entirely on these animal models of disease that are contrived and unrealistic. So to give you one example, the Alzheimer's field has been failing for decades because they are using animal models that are unrealistic to the human case. They take a mouse and they engineer it to produce 10 times the normal amount of a single protein called amyloid beta. And amyloid beta, it does cause some rare genetic forms of Alzheimer's, but 99% of Alzheimer's, it's not apparently the causal factor. And so pharma spent billions, wasted billions, and NIH has wasted billions on this dogma. And that's how science works. It's built on dogmas, and then those dogmas are overturned, that the Earth is the center of the galaxy, the Copernican dogma or revolution, and so on. So anyway, this is how science works, the overturning of dogmas. And the main issue is that pharma relies on these animal models of disease that are not realistic. And so one of the key offerings of the LongBio field is we're using an uncontrived, naturally occurring process of aging. And the aging process looks very similar when you look at yeast, worms, flies, mice, et cetera, and humans. So we think that the success rate in the clinic is going to be a lot higher as a result of that. And we're following that model at ImmuneAGE, of course.
So my last question, and our time is up for today, is that as an investor, I think, and a subject matter expert researcher, you have a very strong feel of it. And certainly you would appreciate the fact that other investors are going to be thinking through risk in terms of time horizon, right? So how long do they have to wait? How many clinical cycles before they can actually de-risk some of these things and truly prove out some of this, some of these theories? How do you, aside from what you just shared, what are other kind of de-risking factors that you can talk about that either shortens the time horizon or shortens or reduces the capital runway so that we can see the results, ideally in a human clinical sooner than later?
For sure. Yeah. So a lot of ways to answer that question. I mean, first, I would say the good news, the headline news is biotech has actually had higher returns and a lower loss ratio than tech over the last 10 years. It was helped by recent bubbleiciousness in the last probably 2 years or so, but still, even on a longer term basis, biotech therapeutics, at least, are outperforming traditional tech. And my personal theory about that is because a lot of the low-hanging fruit in traditional software has been picked, and there's too much capital in that space. So it's, when you get too much capital and not enough good ideas or operators, the returns suffer. Whereas in biotech, in deep tech, and therapeutics, especially aging, which is a very new field, the returns are still significant. And so the drawback with biotech relative to tech, one of the drawbacks is the longer time horizon. So, it takes from, phase 1 to approval, something like 7 years on average. That said, investors do not need to wait until FDA approval to get an exit, a liquidity event. A lot of companies can IPO when they're in phase 2, or in some cases earlier, and out-license and partner with pharma earlier and receive a great return there. So you don't need to wait the whole clinical development timeline in order to get an exit. So as far as other methods of de-risking, I mean, clinical trials are getting a little more efficient. There are sort of digital health applications to reduce the cost of clinical trials. But there's still that fundamental limit, which is the pace of progression of a human disease. And this is one reason why pharma really likes rapidly advancing diseases and rare genetic diseases, where there are some perks to pursuing those indications. But you're really rewarded in terms of returns for the wait, because if you have a drug that works for a disease, for example, a blockbuster anti-TNF antibodies are selling for $20 billion a year for 10 years, $200 billion in returns. That's pretty good for probably all in a couple $100 million invested. So yeah, it's definitely a power law distribution in this space, and it's also for those who are interested in meaningfully contributing to technological advancement. There's also that perk.
So I did say that I was going to ask my last question, but I have another one.
Sure.
And it's a bit of an off-topic question, really, is that we are seeing quite a bit of R&D within the aging in space. And part of that is because of the fact that once you're in space, the DNA is exposed to quite a bit of radiation. So there's a rapid aging of cells. So there's a lot of longevity and aging-related type of experiments that are just beginning in space, oftentimes kind of in a closed shell environment. Space Pharma is one example out of Israel. Others are more done through the typical ISS astronauts and experiments. How could space experimentation could help de-risk some of these things and be able to get some results sooner?
Sure. Yeah. So if we want to do any kind of long distance space travel, we're going to need pretty radical interventions in the damage that's done by space. And it looks a lot like the damage that occurs with aging. So like you said, there's radiation causes DNA damage and inflammation. There's also the microgravity problem. For some reason, the gravitational fields are important to cells. So we've come across a handful of companies with Healthspan Capital looking into this space, if you will. And it could, in principle, accelerate interventions testing. So if you bring animals into space, that could be attractive. But we have progeria, fast aging models, but it could just be another sort of tool in the toolbox to look for interventions. I think the most important aspect of the space field for longevity, the LongBio field, is government interest. So NASA, private space companies like SpaceX, or just other governments will fund this area of research because it has that double effect of not just for longevity, but also for astronauts. So to the extent that we can draw that parallel in the minds of funders and regulators, the better.
Mm-hmm. Great. So with that, I have been joined by Sebastian Brunemeier, General Partner at Healthspan Capital and CEO of ImmuneAGE Pharma. Thanks for joining today.
Thank you. Great to be here.
If you've enjoyed this episode, take a moment to rate our show on any podcast platform that you listen to. Scroll down to the bottom and push 5 stars. It's that easy. And as always, thanks for listening.